Hi, everybody. Thanks again for coming to our latest webinar here. Today, we're happy to have Lynn Jones, who will be talking about, the title of his talk will be the covalent modulation of cereblon using sulfonyl exchange chemistry. So Lynn received his PhD in synthetic organic chemistry from the University of Nottingham before doing some postdoctoral work here in the States at Scripps Research Institute in chemical biology. He then went back to the UK to join Pfizer as a medicinal chemistry team leader and eventually became the director of chemical biology and lead discovery technologies. Staying within Pfizer, he transferred here to Cambridge and was there the head of rare disease chemistry and head of chemical biology as well. From there, he moved to Dana-Therapeutics as the head of chemistry and chemical biology. And now his current role is as the director for the Center of Protein Degradation here at the Dana-Farber Cancer Institute. He's also one of our advisors for this webinar committee. So we very much thank him for all of his input and support throughout this webinar. And his research interests include the use of chemoproteomics and chemogenomics to advance new therapeutic modalities. So we're interested to see how that plays into your talk. So with that, I will hand the floor over to you, Lynn.
Thank you very much, Brianna, and thank you for the kind introduction and the invite. Even though I help out, I didn't select myself for this one, but thank you once again for the opportunity to present our research and some of our research in the center in the area of targeted protein degradation. And so we're a drug discovery group. That's what we do. That's our focus. Rational design and development of degraders and molecular glues. Those working in drug discovery will be very familiar with content of this slide, which really is trying to get an understanding why we fail in drug discovery, where we fail in clinical trials in phase 2. Usually we fail in phase 2. Many sort of analyses and reviews and opinion pieces. Here's one from a colleague of mine at Pfizer, Mark Bunnage, now at Vertex, analyzing, this sort of drop off in success rates through clinical trials. That phase 2 through phase 2, most things fail. And so the reason for that failure is usually lack of efficacy. So most things fail because they don't work. So you might pass through phase 1 where you're trying to understand safety, and then in phase 2, usually you're testing the molecule, the mechanism, the therapeutic hypothesis for the first time in patients. Not only do you see lack of efficacy, but often, unfortunately, there's a lack of PK/PD understanding. So the relationship between the pharmacokinetics of the molecule in humans and then the pharmacodynamic effect. And so, so often, unfortunately, we choose the wrong target, or maybe the right target in the wrong indication, maybe a bit of both. And then what's really disappointing really in this space is that we might go to the trouble of running a clinical experiment and it might fail and we don't know why it's failed. And so many years ago now in Pfizer in the UK, around about 15 years ago now, myself and Mark decided to create a chemical biology group where we had this sort of idea to use chemistry and chemical biology to really get at this, this attrition in phase 2. And we created, for example, a framework published here a few years ago now, a framework to help really the community validate new targets, therapeutic targets, all the way from exploratory biology, the earliest phases of exploratory research, all the way through preclinical in vivo studies, and then ultimately into the clinical experiment. That built off some seminal work really by Pete van der Graaf of Pfizer and Sandwich at the time. Really, our goals have been to, over the years, to use chemistry and chemical biology to advance target discovery, modality discovery, so really broaden targetable, druggable space, and also technologies that sort of shine a light on translational pharmacology in this aspect of PK/PD. I've included then a plot as data from a recent publication from Pfizer. So these are data from Pfizer's clinical trials over the last 10 years, or 2010 to 2020, showing maybe closing the gap on phase 2 success rates, which is great, and maybe a reflection of the work that has been happening across chemical biology and related disciplines, but nevertheless still a major issue that we all face in drug discovery. And so, as I said, the goals, and really have always been the goals in my groups in chemical biology, have been to try to advance new modalities and targets and develop our understanding of PK/PD. And so the focus of this presentation is summarized here. Really, we decided pretty early on to focus our efforts. I'm a synthetic organic chemist by training, as Brianna said, yes. So coming at this from a very sort of synthetic chemistry mindset of using covalent methods, covalent drug discovery, to not only in their own right as drug modalities, so with all the advantages they can bring from dynamic duration, improvements in potency, enhanced selectivity, but also ways to advance alternative modalities. I call it synthetic re-engineering of proteins. I'll come back to this through the talk, but also advancing biotherapeutics and synthetic vaccines. Also then developing new technologies. Chemoproteomics is one that we've really focused on over the years, and I'll touch on that. And then occupancy biomarkers, really to understand this relationship between target engagement and quantifying that effect, and how that relates then to functional pharmacological effects and efficacy. And then the focus of this talk is then the chemistries that we've used on targeting cysteine, really just to show how we started out on, in this field originally, sort of cysteine targeting platform, but then very briefly touching on that and how then it evolved into going beyond cysteine and the use of sulfonyl exchange chemistry and chemical biology to really address the goals that I've highlighted on this slide. So briefly then on cysteine targeting, this is an area that many will be familiar with. It's something that's been covered really nicely in the TBD webinar series, by Dan Nomura, for example, and really. As I said a long time ago now, we developed this sort of platform approach for cysteine targeting in our group in Pfizer in the UK. And on the left of the slide, just one example of how we thought about sort of harnessing cysteine targeting chemistry and chemical biology in the kinase field. And so this is a collaboration with Nathaniel Gray at Harvard at the time, was really to map out reactive cysteines in the ATP binding site of kinases, because each cysteine within that site represents an opportunity to develop a covalent probe, a covalent drug, with the advantages that I sort of highlighted previously in terms of pharmacodynamic duration, development of occupancy biomarkers. And so this is work by Yogesh Sabnis at the time to map out these targetable cysteine residues. And then that was the chemistry side of this. And then we had biological sort of analyses, bioinformatics, then to really bring these two things together to say, okay, here's a really interesting target as compelling disease biology and human biology, and then something that we think that we can drug using the covalent modality. So there's a lot of opportunities even within the kinase space. And so work that came out of this effort, and then that was really led by Pfizer in Cambridge by Abby Torrenson and a medicinal chemist on this project, took one of these targetable cysteine residues in a protein JAK3, so inflammatory target. We wanted compounds that had selectivity over the other JAK enzymes, the other three enzymes within this sort of subfamily, that do not possess a targetable cysteine residue, and that's super important. And so what the team were able to do then was design acrylamides that target this cysteine within JAK3, and work in my group, working with this team, was then to develop occupancy biomarkers to then establish, as I said, this relationship between target engagement and what levels of target engagement can you achieve to drive a functional effect. Other work that came out of the group was to understand selectivity. And that's the beauty of these sort of modalities, that you can convert these molecules into clickable probes. And so the molecule shown here is basically the clinical candidate now with a click reporter, silent reporter, if you like, that allows the molecule then to take a snapshot of occupancy in live cells, in primary cells, and then click on biotin immunoblot for your targets of interest. And through this effort, really, really important effort that led to the appreciation, really, that this molecule, ritlecitinib, is not just a JAK3 inhibitor, but also engages the other proteins in this class that possess the same reactive cysteine residue. So if you like, a rebranding of this molecule from being JAK3 to a JAK3 tech selective molecule. So cysteine has a lot of advantages, and when you think of this from a platform perspective, why target cysteine? And it's highly nucleophilic. It could be PK perturbed in an enzyme binding site that makes it even more reactive. It's a thiolate. There's a great toolkit of electrophilic warheads in terms of chemistry. You target that residue, and you can often mutate cysteine to a serine in particular that can confirm binding site selectivity and reactivity with that site. And so this is a common trick used in target validation experiments, where you then see significantly less potency when you with your molecule, with your covalent cysteine targeted molecule, when you perform that trick. The challenges, though, in this field are really the flip side of what's on the left there. That's that high nucleophilicity can hamper the development of really selective molecules, maybe illustrated a little bit by ritlecitinib on the previous slide. Probably the most important and really important to this presentation is that most protein binding sites do not contain reactive cysteine. Okay, and then this cysteine serine mutation, whilst being a cool trick that you can use in target validation, can lead to mutational resilience in the clinic, in the cancer field in particular. And so several years ago now, we came at this again from that sort of platform perspective and saying, well, what can we do then to go beyond cysteine for these reasons? And that led us to really building out and exploring sulfonyl exchange chemistry and sulfur 6 fluoride exchange chemistry, such as sulfonyl fluorides, and that'll be the focus of this talk. So really, though, we've built on the pioneering work of many folks who've worked in this field over the years, but two in particular that I wanted to highlight. So Bernard Baker and Roberta Coleman. So Bernard Baker, medicinal chemist, back in the 60s, really was the first to incorporate this sulfonyl fluoride warhead, and I'll talk more about the chemistry, the sulfonyl fluoride electrophilic warhead into reversible binding ligands for proteins to convert them into irreversible covalent inhibitors and protein modulators. So, but yeah, the first guy to really do this, and then Roberta Coleman built on this to incorporate this warhead into metabolites. And so, for example, here, adenosine mimic FSBA, so fluorosulfonyl benzoyl adenosine. And the idea here was to map out nucleophilic residues in metabolite binding sites in proteins. So, you know, really elegant biochemistry work using the power of covalent organic chemistry. And so what was shown over the years by us and by other groups is that the sulfonyl fluoride warhead, the electrophilic warhead, can engage a number of amino acid side chain residues. So tyrosine in particular, but also lysine, serine, threonine, histidine, all serendipitous discoveries, not by design, but shown later after the fact. And so work by ourselves, really important work by Barry Sharpless and G. Finn, sort of exploring the sort of chemistry side and the opportunities of sulfonyl fluoride exchange chemistry. That's really sort of reignited this field and how it can be used, particularly in drug discovery and chemical biology. And just a few words on the chemistry of sulfonyl fluorides in particular. They sort of have this Goldilocks reactivity. They're remarkably stable in aqueous systems, in waters, in cells. They tend only to react in protein binding sites, like any chemical reaction. Yeah, you can force it. It can react non-discriminately with lots of different things, but you know, the chemistry is really quite discerning. It's quite privileged, and so you tend to get this sort of templated reaction within protein binding sites, illustrated on the right here in this cartoon that we published a couple of years ago with Jeff Kelly, just illustrating the sort of specificity of this chemistry. And so, yeah, good water stability, minimal perturbation of lipophilicity. And so we've incorporated this warhead deliberately over the years to target these residues. And just a couple of slides on how we've used this. Here's an example of rationally targeting a tyrosine residue. It was the first time, I believe, that had been done. So these were molecules that were discovered in a phenotypic screen for looking for regulators of survival motor neuron protein, which is deficient in spinal muscular atrophy. And so these molecules, a putative target was identified as the decapin scavenger enzyme, an enzyme involved in mRNA metabolism. And so we wanted to validate this target. We wanted to validate it in human primary cells. And we want to understand the relationship between target engagement and phenotypic effects. And so we targeted a tyrosine residue in the binding site. We converted these molecules into a click reporter of DCPS engagement, validated the target in cells, and built out PK/PD understanding for this, really providing us then with the confidence to move forward to optimize these molecules into now reversible binding, super potent, very efficient inhibitors of DCPS, then validated this mode of action in mouse models of the disease. And so this was work led by Malik Abalsami and MedChem in my group. All the chemical biology that I've described thus far, and these stories as well, was by Eric Hett and Hua Zhu in my group at the time. And then just another example of tyrosine targeting. This is on the MedChem side, was work led by Les Dakin at Pfizer at the time. And so these were peptides that were inhibitors of IL-17, anti-inflammatory target. We didn't know exactly how they bound. We didn't have a structure at the time. We had docking models. And these molecules were not particularly potent, metabolically unstable, as you might expect from linear peptides. And so we wanted to understand the binding site. We incorporated sulfonyl fluorides from the docking model. We thought there was a potential for a proximal tyrosine residue. showed that these sulfonyl fluorides engage that tyrosine, give us confidence on the model. And you can see that the molecule binds in this sort of horseshoe shape. And so that inspired Les then to cyclize these molecules and make them a lot more potent and metabolically stable. And then we've targeted lysines. This was a collaboration with Jack Taunton and Al Burlingham at UCSF to develop reporters in live cells for the first time now of kinase ADB binding sites. So we use computational proteomics to determine selectivity in live cells using this probe XO44 that is designed to engage the conserved lysine. And there's a commercial probe. We can use this for determining kinase selectivity. Something more selective, and again, you can regularly do this. Using structure-based design, incorporate sulfonyl fluorides to create reporters of occupancy. Ilotinib comes with its sort of inbuilt click handle, which is always quite handy. And then going back to DCPS then, and I'll come back to the fluorosulfate chemistry, intrinsically a lot less reactive than sulfonyl fluoride. And what was interesting when we incorporated this warhead into these DCPS inhibitors is that now the warhead now flips down to engage a non-catalytic serine residue in the binding site. And so this is one of the few examples beyond aspirin where we can engage a non-catalytic serine. We've used this chemistry over the years in all sorts of different ways to stabilize membrane proteins for structural biology, mapping binding sites, cryptic sites, metabolite binding sites, target ID from phenotypic screens, and others I don't have time to go into. So we're going our way through the amino acids, right? So what's next? So histidine. So we thought, well, what about histidine? Histidine's pretty underexploited, I would say, because when you look at histidine, it's a pretty special amino acid residue. If you look at the measured pKa, this is a great database, PCAD. Urge you all to, if you're interested in this sort of stuff, I love this stuff, have a look at this database that was published a couple of years back. Measured pKa's of amino acid residues in proteins. Histidine is very, very rarely protonated. It's usually unprotonated, unlike lysine, for example, which really can impact the ability to target the residue with electrophiles. Low surface frequency, it has the second highest preference to be within striking distance of drugs and drug-like molecules. It's the most frequent in enzyme binding sites, shown here on the right. It's in particular enriched in oligo binding sites in proteins as well, certainly more than cysteine, as you'd expect. And it's often a catalytic residue. It's actually there to be nucleophilic in terms of its biochemistry. So anyway, some of the background on why I think histidine's a really interesting residue to target. Some examples in the literature of serendipitous labeling of histidine, and I go back to this probe from Roberta Coleman, back in the day, this FSBA, that others have shown can label histidine. Two examples. So this one at the top in bovine mitochondrial ADPAs, and you sort of get really interesting, actually, you get sort of pKa dependent labeling of either a tyrosine residue at higher pH and then a lower pH of the histidine in the same site, which is really interesting. And then other work by Robert Switzer was observed labeling within PRPP synthetase by FSBA. And actually, it's really interesting. If you look at the crystal structure, it sort of prefers to label a histidine as opposed to two proximal lysines in that binding site, and maybe for the reasons that I explained in the previous slide. I put this example in because this is pretty relevant to this audience, and I'm sure many people are familiar with this story. In particular, Craig Crews, of course. So ovalocin was found, really, really nice work by John Clardy, Jun Yu, Craig Crews, as I said, to show that ovalocin and fumagillin, so these natural products that contain a spiroepoxide now as the natural electrophilic residue in the molecule engages a histidine in methionine aminopeptidase 2 protein, shown here as a crystal structure shown that engages this spiroepoxide. And so what Craig Crews and Ray Deshay did, is off the back of this, create then the very first PROTAC molecule. So converting ovalocin synthetically through to a molecule that is then linked to a phospho binding element for SEF E3 ligase. So this was the very first PROTAC, and it's interesting, the very first PROTAC was a covalent histidine targeting PROTAC, but on the protein of interest side, not the E3 ligase side. And I put this example in because it is an example of rational design of histidine cross-linking using sulfonyl exchange chemistry, but not using a small molecule. But this is really elegant work by Lei Wang at UCSF, shown proof of principle, right, with protein cross-linking. And so two proteins that interact, so protein Z and its corresponding affibody. There's in the crystal structure, you can see a salt bridge. And so what the group did was convert this residue into the fluorosulfate version of tyrosine, and show that you can then cross-link when you mutate this to a histidine residue, shown by peptide mapping experiments. So shown proof of principle. But the question was, can you do this with a small molecule? And so I'm not going to go into a lot of detail on the what's on this slide. I think most people are familiar with the fascinating story of the mode of action of the amide drugs. An example shown here, a structure shown here in the middle from Eric Fisher here at Dana-Farber, of course, and glutama, as an example of showing that the amides bind in this site within cereblon that enables then the recruitment of a new substrate, again illustrated purposes, CK1 alpha, to collocate then that target with the ubiquitin machinery, polyubiquitination and proteasomal degradation of the target. And so we had this idea that maybe you could covalently engage a residue at the surface of cereblon, and maybe you would do one of two things, right? Maybe you would completely abrogate the ability of the complex to recruit a new substrate, or it might subtly change the surface of cereblon where you now recruit maybe different substrates. That was the idea. And when you look at this particular binding site of cereblon, there isn't a cysteine. In fact, the only residue nucleophilic that we thought might be targetable was histidine 353. So we thought, let's make some sulfonyl fluoride and fluorosulfate containing probes to engage that histidine, and then screen it and see what it actually did to the function of cereblon. I should point out here at this stage that some unpublished work, I think in a patent from Vividian and Ben Cravata, shown, there's cysteines actually elsewhere in cereblon that can be engaged, that then have an allosteric effect on the ability to bind and recruit new substrate proteins. So they tend to inhibit the binding of the amides and molecular glues in this site. So, but we went in a different direction. We're going to target the specifically surface residues. So I got all nostalgic on this one. I thought I've got previous in this space, and this was an amazing experience during my bachelor's degree. Whilst at the University of Bath, I did an industrial trainee year, working on molecular glues. I didn't know it at the time, but I was working on molecular glues. This is nearly 30 years ago, in the lab of Richard Sidrani, who's still at Novartis, who was Sandoz Pharma back then. So here's a picture of me with Richard and Bin Tai in the lab in 1993. And we were working on rapamycin. So, very early molecular glue research. Obviously, Stuart Schreiber has, sort of pioneered this sort of research with others, but used the term molecular glue for this type of molecule. And at the time, we knew that the molecule bound through this binding component, FKBP12, now FKBP12, and we knew that this region of the molecule was, we call the effector element. We knew that you could bind FKBP12, but you needed this component then to achieve the phenotypic effect of the molecule, immunosuppression at this time. But through research and amazing research out of Richard's work, then led to everolimus, a drug used for treating cancer and other diseases, and actually came from modifying this cyclohexanol. At the time, my work was to further explore the pharmacology of this region of the molecule. I'm not going to go into the synthetic chemistry, but it was really fun and complex and challenging, but really, really interesting. And the idea was, can you modify this group and see how it would affect its binding and its effector function? As we made quite a drastic change for this chemistry through to the cyclohexanol group that we then eventually published a few years later, showing that these molecules, they still bound to FKBP12, could not then recruit, what was shown later, could not recruit mTOR as the other partner, and then crystal structure work. John Clardy's Schreiber showed that the cyclohexanol motif just sits at the interface between the two proteins, and so this idea of mucking about with parts of the molecule at the surface of a protein to perturb protein-protein interactions is something that I'm really, really just naturally interested in because of that previous experience. And so just a little bit more on the chemistry, if I may, because we won't be able to do any of this work without the synthetic chemistry, and not as maybe as complex as the chemistry on the previous slide, but nevertheless really important advances made by Barry Sharpless and other groups, our group as well, made important contributions here to now make this chemistry really amenable to the community. We converted bromo EM12, so one of the imids, so it's commercially available, incorporate the benzyl sulfide and then oxidative chlorination and then conversion of the sulfonyl fluoride to the using potassium fluoride to create this probe with this electrophilic warhead to engage that, designed to engage that histidine 353. This chemistry worked great. You get 50% overall yield, works really well. And then if you like click chemistry, conversion of phenols in one step to fluorosulfates, really, really useful transformations. We can do this in one step. And this has been advanced by several groups now. And in particular, I'll just point out Chris and Andy at Pfizer developed this really nice reagent that allows you to do this simply. So that's on the chemistry side. Now on the biology and how do we screen these molecules. This is work out of CPD, actually by Brianna as well, who took the lead here with Radek Novak. So how do we measure binding and potency? We believe in the CBD, we really want to understand target engagement in live cells, and then that translates to pharmacology. And so here we developed using the Promega technology, nanobreath technology, an in-cell target engagement assay. And so if you're not familiar with this technology, you basically take your target of interest, you fuse on nanoluc luciferase, and then you set up a breath signal with your drug tracer. And so in this case, we developed sort of BODIPI labeled lenalidomide. And this molecule, maybe remarkably based on its physical chemistry, this works really well in live cells. And so we can use this as the tracer, and then you just compete out the signal illustrated here using EM12. Promega themselves actually have reported a similar approach. This is actually a stable cell line, and the structure of the probe is given here, which I think can help the community. And we're in the process. I think we've just submitted this one for publication. So anyway, so we have the chemistry, we have the screen. How do these compounds look? So we incorporate sulfonyl fluoride, as I said, into this EM12 template. I should say we chose EM12 just to show proof of principle, and the chemistry is a little bit more tricky when you have the extra amino group that you see in pomalidomide and lenalidomide. So we chose something a little bit simpler in terms of the chemistry. And then we look at the potency in the nanobret in cell assay. You know, obviously this is a single time point. We see time-dependent inhibition, as you might expect to see for a covalent inhibitor. But you know, the single time point, I think at 2 hours, you know, extremely potent inhibitor or binder, you know, of cereblon. You move the electrophile around one position, and as you'd expect, based on the structure, if you recall on the previous slides, you lose significant potency. Nevertheless, though, still actually really potent ligand of cereblon. We converted the sulfonyl fluoride, quite nice chemistry, converted this into the sulfonyl triazole. So this is inspiration now from, you know, Ken Xu at UVA, who's done some really nice work on sulfonyl triazole exchange chemistry. And so we thought, okay, we'll give that a go as well. So the 1, 2, 3 and this isomer, again, retaining potency on cereblon in this assay. We then converted the electrophilic warhead into a fluorosulfate. As I said, this is intrinsically a lot less reactive. There's extra oxygen atom, then makes the sulfur electrophile less reactive, but nevertheless retains potency on cereblon. And then, as you'd expect, you move it around one position, you lose significant potency, as I showed you with these molecules. And so I'm just going to I'll talk about all these molecules, but I'll focus on the sulfonyl fluoride, EM12 SO2F, as we call it, and the fluorosulfate EM12FS in the coming slides. Okay, so first thing, do you label the protein? If you look at the sulfonyl fluoride, yes, it labels really nicely. It labels once. You take the cereblon DB1 complex in mass spec, you see a mass shift that corresponds to sulfonation of the protein. You don't get any labeling of DB1, so it's selective. We also use BSA, so bovine serum albumin, just to show that you get selective labeling of your protein of interest. Similarly for EM12FS, or the fluorosulfate, again, you get a corresponding mass shift. As you'd expect, it labels once, no labeling of DB1, no labeling of BSA. And now using peptide mapping, we can show categorically the molecule labels histidine 353 and only histidine 353. And I should say this work was led by Justin Crude in the CPD and Scott Ficarra at Dana-Farber. We looked at the other electrophilic probes, and they behave very similarly. Yeah, so you can label quite nicely histidine 353 using these other probes to greater or lesser extent, as I said, based on the SAR and how well they're templated to react with that residue. Okay, so what do these molecules do? So one way you can look at function is degradation proteomics, is work by Rebecca Mativier and Catherine Donovan, along with Jeff Dan at the CPD. And so what's really interesting is that these molecules now, these sulfonyl fluorides or triazoles, they actually don't have any neosubstrate degradation activity. So, or if they do, it's really, really weak, say on CK1 alpha, GSP1, but nothing really to write home about. Okay, so they generally inhibit the function. And so we thought, well, that could be quite useful because you might then be able to use this type of probe in target validation experiments. So, where you might want to invoke a mechanism of degradation that occurs via cereblon. You might use genetically engineered cell lines. You might use, the degraders of cereblon itself, where there's a time component in terms of how quickly you can degrade. So this is orthogonal to those methods and works really, really well. So just proof of principle here from Jeff is that you can take cells, lenalidomide as an image can degrade IKZF1, shown here as one of the neosubstrates, zinc finger transcription factor, and then you can completely inhibit this then using the sulfonyl fluoride probe. So we've used this in multiple experiments, works really well, and again, could be a useful tool for the community. So the fluorosulfate, okay, so in degradation proteomics, and I should say this experiment is, you take cells, incubate, in this case 1 micromolar, 5 hours, and then look for changes using quantitative mass spec proteomics in the fold change of proteins. So this axis is significance in terms of p-value, and then this one is the log fold change. And so we're looking for proteins, in the sort of bottom left corner, and we look at the fluorosulfate, we see one protein that is robustly down-regulated in TAC1. I'll talk a little bit more about TAC1, but yeah, just this one protein in TAC1, remarkable result in selectivity. We see it across different cell lines. I've shown 2 here, validated in Western blot. We can inhibit it using modulation inhibitors. So this is a really interesting result and a new substrate, a new target that we had really set out to do originally. Can we degrade something really quite different? And that seemed to have panned out. So biochemical work now from Justin really to dive into the mode of action at more of a molecular level using these acids, really elegant work actually. So set out the TR fret between cereblon and NTAC1, and showing that you get this increase in signal, which is time dependent, that perfectly overlaps then with the rate of forming the covalent bond between the probe and histidine 353. Okay, so very strong evidence that it's not just the binding of this molecule to cereblon that causes the downregulation of NTAC1, the degradation of NTAC1, but it's also the covalent engagement of that residue, which is special, that results in that effect. I'm not going to go into, in the interest of time, I'm not going to go into detail on NTAC1. There are no chemical probes for NTAC1. It's deemed undruggable, if you believe such things. So what is NTAC1? It's actually involved in degradation itself. It's a pretty interesting result, right? So it's involved in the NN rule pathway. It's N-terminal glutamine hydrolase. So it's involved in this step that then would lead to arginine transferase activity, and then this recognition of this by other E3 ligases and degradation. Okay, so this is really nice work from Vashavsky and Huang, showing that USP1 actually is a substrate for this pathway. So USP1, a deubiquitinase fragment of USP1, is a substrate of NTAC1, and this is actually involved in deubiquitinating DNA processivity factor PCNA. And so it's sort of interesting potential therapeutic approach here by inhibiting and degrading NTAC1. It might give you an opportunity to control the DNA damage response. I get asked a lot about these warheads in terms of their suitability for drug discovery, and so I'll cover that in the coming slides. So just as reference point, EM12 here shown at the top in terms of its human plasma stability. So just focusing on plasma stability. And as you might know, the immunomodulatory drugs, the IMIDs, with the glutaramide, they tend to have some turnover in human plasma anyway, sort of within that motif. And so when you incorporate the sulfonyl fluoride and triazoles, you see really low stability in plasma, which is not actually surprising, for this particular motif. So generally low stability. It would make that therefore quite tricky to convert that into a drug. But of course, there's always a way in chemistry. But this is perfectly suitable for chemical probes, and so we've used this chemistry over the years and created a lot of high value chemical probes with utility in drug discovery. At the bottom here, the fluorosulfate, on the other hand, is rock stable. So this is really at the other end of intrinsic reactivity and metabolic stability. And so, yeah, the fluorosulfate is a very stable motif, and certainly suitable for drug design. And then just to drive that home on this slide, if you compare EM12 versus the fluorosulfate, just a bit more data on suitability for properties in terms of drug design. You really do maintain that excellent metabolic stability, don't affect permeability or solubility to any great extent. So it's sort of really useful latent electrophile. And going back to the DCPS, the decoupling scavenger enzyme inhibitors, again, you can incorporate this warhead, this work that we've published, and still retain, good properties in the molecule and converting these molecules though into covalent modulators. So yeah, so just to summarize, the story here is by making this very, very small change in the electrophile, just by adding one atom, we go from being a molecule that is essentially very useful as a sort of inhibitor of cereblon, blocking the cereblon through my binding site, and then just adding that one atom can now convert the molecule into a degrader of NTAC1. Here's some docking, covalent docking from John Chi in the group, just showing, when you look at the structure, this is now lenalidomide with C7 alpha, so going back to that original structure. And if you look at the Histidine 353, by design, targeting this residue, which is proximal to the imids, it sort of resides on this loop. This has been called like the sensor loop recently by Gabriel Lander at Scripps. So this loop sort of flips up when the imid binds, but nevertheless, this histidine residue, which would normally be right at the interface, with a neosubstrate, and is actually making interactions between cereblon and that substrate. It's really interesting is that in the docking model, you see this slight flip of the residue towards then the electrophile, and then the sulfonyl oxygen atoms in this region really sort of clash with the standard substrate in our knowledge of Degron recruitment. So sort of explaining, maybe why we don't see significant degradation of the usual substrates. And clearly there's something funky going on with NTAC1 in terms of its recruitment and degradation requires, more research to understand that. So just running out this presentation then, I mean, it sort of leads you also to consider what's special on the protein side. So this residue, this histidine 353, I made maybe a case for it to be targeted rationally in terms of drug design and chemical probe development, but it's still relatively sort of unreactive, particularly compared to cysteine. So what's special about this residue? I mean, we're not the only ones to show that you can engage this residue. And this is work by Christina Wu, who recently reported a photo affinity probe based on lenalidomide that incorporates a diazarine, and that on photo affinity labeling is shown to cross-link to that same histidine. Yeah? So that might actually be via the diazo species, the work that we did with Christina Wu in collaboration, showing deeper dive on the mechanism of how diazarine photo cross-linking works and its mechanism. And so it's interesting that maybe histidine 353 is cross-linking through that type of mechanism. This residue and this region is very well conserved across species, and sulfonation of a histidine residue is not a million miles away from maybe phosphorylation. We've shown that these sulfonyl fluoride probes can act in that manner and label residues that would ordinarily be phosphorylated. And so it sort of leads to an interesting sort of hypothesis that maybe this histidine actually is quite special, either in terms of a PTM control mechanism of cereblon itself and its endogenous function, but that would require more work to understand that. And so, yeah, I mean, we've heard a lot about the opportunities for cysteine. There's still a huge uncharted space there with regards to cysteine. Really nice sort of perspective from Dana-Farber in this space. Just looking at all the E3 ligases and the target cysteines in those proteins, and some of which are shown here from a publication on the right. What I would say through work that we've done, this work that now we've shown maybe with cereblon, is that I would say there's even more opportunity in the chemical biology arena to target residues beyond cysteine. So how can you use this chemistry apart from covalent molecular glues to create new substrates? You could imagine developing covalent catalytic degraders maybe more effectively by targeting these residues at the surface of E3s and the adapted proteins. And so, for example, making covalent PROTACs, but now covalent on the E3 ligase end, not on the protein of interest end. So truly, maybe enabling the catalytic function, and we've shown some proof of principle there with a PROTAC molecule. Site-selective protein surface reengineering, and there's loads and loads of ways that can be used and harnessed even beyond protein degradation. And then neofunctionalization, so chemical mutagenesis of surface residues could then impart new functions on proteins in cells, maybe in a sort of a synthetic biology way of thinking, using the power of synthetic chemistry in live cells. And so, yeah, I just wanted to now acknowledge the efforts really of the entire group at the Center for Protein Degradation here at Dana-Farber. In particular, the work of Justin, Jeff, Brianna, Radek, Fidel, in terms of the biochemistry story behind the elucidation really of the mechanism of these covalent cereblon modulators. Catherine, Rebecca, Scott on the mass spec side. In particular, also my colleagues and friends and collaborators, Ben Ebert, Eric Fisher, Nathaniel Gray, within Dana-Farber and Stanford now, funding from Deerfield. I also point you to our website. We're in the process of enhancing our website, but please do check us out. We actually have 2 open positions in the group currently. So if you're interested in sort of really advancing, new therapeutic modalities for drug discovery, then please check out these positions, one in biochemistry and one in chemical biology in our group. And finally, I'd like to thank you for listening.